So I guess it's no need to present you who you are, and you are going to speak. Thank you very much about this fact that you're going to speak about current gene therapeutic efforts in terms of an overview. Thank you very much, Bernhard.
You're welcome. So it's my pleasure to discuss this theme with you. The way I would like to do this is that I would like to remind you of the promise of gene therapy in HD, then discuss and describe to you the landscape as it presents itself this year and the various players to the extent that this information is in the public domain. Then I will discuss with you the challenges for gene therapy in HD and close with a look in the future. A look into the future to discuss improving a precise in vivo targeting of vector-based therapies, ensuring a high transduction efficacy and various approaches to minimize the immune-mediated reactions towards vectors and their cargo. So let's start with the promise. From my perspective, the promise is amazing. Fixing the root cause of monogenetic disorders is one of the promises. If we are looking at autosomal recessive or X-linked disorders by supplying a missing or faulty gene product. If we are looking at an autosomal dominant disorder with a gain of function mechanism, mostly responsible for the symptoms and signs, at least according to our current understanding, like in Huntington's disease, the purpose is to silence and to suppress the mutant allele. And then there is the promise of gene editing, that is to correct the genetic defect at the DNA level. And to start with, that would apply to all monogenetic disorders in principle. The second big promise that comes with gene therapy is that potentially one treatment session may last for a life. So it's a one-time only treatment. Whether that's actually true needs to be established, but likely it's a long-lasting treatment effect, if there is one. So if gene therapy would have been available, for instance, and the last Tsarevich of Russia, who suffered from hemophilia A, here is his picture, and his parents would have been in no need of Rasputin, here is a picture of him, not a very trustworthy person, I guess, and could have been replaced with gene therapy that's still in the works, but here a publication from 2017 describing this working in principle, AAV5 supported. More important for us is that in vivo gene therapy today seems to work also for a CNS disorder. In this case, the SMA type 1, the most severe form of spinal muscular atrophy, where in an AAV9 vector, a functional copy of the deficient gene is infused. That's taken up by neurons, in particular the spinal motor neurons, and it is the cargo is not integrating into the human genome. It's on an episome and is producing the full functional mRNA, including the critical exon 7 required for a full functional form of the SMN1 protein that's ultimately required to keep spinal motor neurons alive. In vivo gene therapy approaches do differ in a couple of respects, and I would like to review this briefly with you. First of all, of course, they differ with respect to the cargo they contain. For instance, the code for therapeutic agents like microRNA, zinc fingers, and various other gene editing instruments. They differ in the vector system employed. for instance, various types of AAVs, adeno-associated viruses, lentiviruses, nanoparticles, potentially in the future. And they differ in the route of administration. Very often it's intraparenchymal injection, for instance, in brain, or a systemic intravenous administration, as I illustrated for the AAV9 therapy for SMA1. Currently, most trials use as a vector the adeno-associated virus, or AAV virus. And here is a picture of a colorful picture of the surface characteristics of this virus. It's a non-pathogenic Dipendo-Parvovirida, and a family member. It has a capsid with no envelope, single-stranded DNA, and very potent, able to transfect post-mitotic cells, in other words, neurons or muscle cells. 13 serotypes are known to exist, and there are more than 100 variants in existence, and it's thought to be non-pathogenic in humans. Currently, and I think the number is pretty impressive, more than 370 human clinical studies are employing AAVs, according to a report published beginning of this year. So let's focus on gene therapy in HD this year and who the players are. So yesterday we heard already about uniQure and their program. There's also Spark Therapeutics, there's Takeda, and there is Voyager Therapeutics, and I'm going to review very briefly what is public information on their programs. Let's start with uniQure. We did hear Dr. Cooper talk about their program to us, two trials, TRX1 and TRX2. It's the first gene therapy approach in HD reaching patients, actually. It's a phase 1, phase 2b for early stage in Huntington's disease. It's a microRNA as a cargo, and it's a non-allele selective approach. And you heard yesterday the therapeutic agent is called AMT-130, and it's an AAV5 serotype, and importantly, it's targeting the exon 1 that does contain the CG expansion mutation as well. And it's supposed to be injected one time intracranially into the parenchyma, and in particular in the caudate and the putamen, in the striatum, in other words, with a catheter and a convection-enhanced delivery system overall. Sparks Therapeutic has a partnership with Roche. Sparks program is at the preclinical IND enabling stage currently, and no public information is at this point in time available as to cargo, vector, and intended route of administration. And that's what you can find on the website. It's candidate optimization and IND enabling. Takeda is very invested in HD. As you may know, they have a partnership with Wave Life Sciences. You heard yesterday about the ASO program of Wave. They have a partnership with Anima to discover mRNA translation modulators, for instance, for HD. So linking to the presentation that Beth just gave and the presentation that Brian from PTC is going to give in a few minutes. And in addition, they acquired Sangamo Shire and their program of an engineered zinc finger protein transcription factor. And overall, the impression is that Takeda appears to home in on allele selective HD silencing approaches. And the gene therapy approach that is currently in preparation at an IND-enabling stage will employ an engineered zinc finger protein transcription factor that does recognize CAGs, and in principle would be a potential intervention for all CAG repeat expansion disorders by silencing the expression of the respective disease-associated gene. And this has been extensively studied preclinically, and I highly recommend you reading and reviewing the publication that CHDI Nacho, in particular Ignacio Munoz San Juan and colleagues of Sangamo have published recently. Voyager Therapeutics did announce that they're going to start a safety and tolerability study with Voyager HTT01 in adults with early manifesting Huntington's disease. Is the study description, and you can find it at clinicaltrials.gov. And the intended start was July 30th. However, on August 16th, they announced that they withdraw their IND and in favor of a second generation program that would enable intravenous administration. So that's what you find on their website. It's currently back to the discovery stage because they have to adjust their tracer AAV capsid. Trace method. Tracer stands for tropism redirected of AAV by cell type specific expression of RNA. That was published as a method working in mice. And in mice, you do find a pretty impressive up to 400-fold higher brain transduction over AAV9 following systemic administration. So that's what this prospect was one of the reasons for Voyager pulling the plug at the last moment. There was also a change in leadership, and the change in leadership had to do with adverse events that was observed in their Parkinson's gene therapy program. So this is what was observed. That's not from this particular study. These are pictures that I owe to Ronald Crystal from Wales and Cornell Medical College, who presented that at a recent FDA advisory hearing. With his program as well as the program of others, and they saw this flare and abnormalities with no clinical correlates that are troublesome. So what are the challenges?
Okay. That's great.
Almost done.
Almost.
Good. And what are the challenges? The challenges are, for instance, toxicological issues. Last week, reviewed in a program that what a hearing by the FDA, vector integration to oncogenicity risks, mostly not applicable was the opinion for humans. Hepatotoxicity, a very real problem with high dosages. Thrombotic microangiopathy, dorsal root ganglia toxicity, mostly observed in animals, and neurotoxicity, the brain findings that I just described to you. So the other challenge obviously is whether all brain areas that you want to cover can be covered. Again, I'm quoting the opinion of Dr. Crystal here using the catheter-based administration technique. You can treat localized CNS disorders, but if you need to treat the entire brain, that's far more challenging. An obvious idea would be to use agents that do cross the blood-brain barrier, like AAV9, that may work under special circumstances, but there are limiting factors for obvious reasons. The number of AAV vectors required may be very high, and eliciting an immune response or other AEs, hepatotoxicity, for instance, difficult to produce at the required purity and obviously costly. If you do this, and Jill published this recently in working with a vector developed by Benjamin Deverman and looking at the transduction efficacy, well, the entire brain can be reached, but the transduction is 10 to 15% of cells, 5 to 15% of neurons. That's the estimate. Then another obvious challenge is once the cells are transfused, you have no longer control over dosage of the therapeutic agent. And I believe for all non-allele selective approaches, it would be of interest to establish the percentage of cells and neurons with no or very low expression levels of Huntington. So what is the future? The future is capsid engineering, where you modify the surface characteristics of the vectors used to allow for precise in vivo targeting crossing the blood-brain barrier, but limiting off-target transduction, for instance, in liver, thus reducing the dose required and therefore dose-dependent adverse effects, optimizing a high transduction efficacy, and escaping pre-existing immunity by modifying the surface. And that's all in the works, but we are not there yet. This is Bertha Benz, the wife of Benz, who came up with the first car, and that's where we are currently.
This is a challenging exercise, like Bernhard. This is definitely a challenging exercise, and I would rather take one of the previous one.
That's fine. I'm done.
Okay, that's great. Thank you very much, Bernhard, indeed.